Welcome to the pod Biomanufacturing and Fermentation Technology. In this episode, we translate microbial science, process engineering, and manufacturing realities into insights that scale.
Welcome in, everyone. If you're joining us today, you're probably the kind of person who likes to, cut through the noise and understand exactly how things work.
Absolutely.
And today's deep dive is taking us straight into a massive, multi-million-dollar bottleneck in the biotech industry. We're looking at why perfectly engineered biological processes, specifically things called fed batch processes, work absolutely flawlessly in a small lab, but then fail spectacularly in a giant factory.
Yeah, it really is one of the most persistent challenges in biomanufacturing right now. I mean, we've engineered these incredible microbes to produce everything from life-saving pharmaceuticals to advanced biofuels.
Right.
But coaxing a microbe to produce a drug in a little glass flask is one thing. Getting that exact same microbe to survive and actually produce at a massive industrial scale, that is a monumental engineering hurdle.
And our mission today is to figure out why that scale-up is so notoriously difficult. We're going to uncover the root causes of these failures, look at a real-world disaster where everything went wrong, and then explore the ingenious control system, the balanced diostat that engineers use to save the day.
It's a fascinating fix.
It really is. Okay, let's unpack this. Imagine you are at home, standing at your stove, making an incredibly delicate hollandaise sauce in a tiny saucepan. You can control the heat instantly.
Yeah, if it gets too hot, you just lift the pan.
Exactly. You can whisk it rapidly, ensuring every single drop of that sauce is perfectly mixed. Now imagine trying to cook that exact same delicate sauce using the exact same recipe, but in an Olympic-sized swimming pool.
You can't just scale up your whisk.
Right. The physics fundamentally change.
That is the perfect analogy to describe what engineers call the illusion of the lab scale. It's a very dangerous trap.
The illusion of the lab scale.
Right, because in a laboratory fed batch process, you're usually working with a small bioreactor. We're talking maybe 1 to 10 liters of liquid. At that tiny scale, the system tricks you into a false sense of security.
How so?
Well, the volume is so small, you have a massive
Surface area to volume ratio.
Exactly. So if the system gets too hot, cooling it down is nearly instantaneous.
Okay, wait, hold on. I get that a small pot cools faster than a giant tank, but how much of a difference does the whisking, the mixing, really make?
Oh, it's night and day. In a typical 5-liter lab setup, your complete mixing time is just 3 to 8 seconds.
3 seconds.
Yeah. That means if you pump in a drop of glucose to feed the cells, it's distributed perfectly uniformly, almost instantly.
Wow.
And because the mixing is so violent, the system has a huge capacity to dissolve oxygen. Engineers measure this oxygen transfer rate using a metric called Kala.
Wow.
Right. Think of it as the system's lung capacity. In the lab, that lung capacity sits comfortably between 400 and 800. It's incredibly high.
So the cells are basically breathing normally, getting all the oxygen and evenly distributed food they want.
Exactly. But then you move to the industrial scale. Massive stainless steel tanks holding 500 to maybe 20,000 liters. Suddenly, all those built-in physical advantages vanish.
Because you're in the swimming pool now.
Right. At 10,000 liters, that metric, the lung capacity, it plummets from that 400 to 800 range down to a sluggish 80 to 250.
That is a massive drop.
Yeah.
It's like trying to breathe through a cocktail straw while running a marathon.
What's fascinating here is the sheer chaos this creates inside the tank. Because of that massive volume, the mixing time goes from a brisk 3 seconds up to anywhere between 30 and 120 seconds.
Wow, up to 2 minutes just to mix.
Yeah, which creates extreme spatial heterogeneity. You end up with dissolved oxygen varying by 10 to 40% from the top of the tank to the bottom.
So a single bacterium just floating through this tank is essentially going through entirely different climate zones.
Precisely. And the same thing happens with their food. Because it takes so long for the liquid to mix, massive glucose gradients form. You get zones right near the feed pump where the glucose concentration is 0.5 to 5 grams per liter higher than the rest of the tank.
So they're getting slammed with food in one spot and starving in another.
Yes, extreme feast and famine conditions. Oh, and heat removal. No longer instantaneous. There's an agonizing 5 to 20-minute lag to cool that much liquid down.
And we can't forget the human element here, right? Because in a lab, you can pull a sample every 30 to 60 minutes, test it, and tweak the feed if the cells look stressed.
Easily.
But in a huge factory tank, labor costs and the terrifying risk of contaminating a multi-million-dollar batch mean you're restricted. You might only be sampling every 2 to 4 hours.
Which means between those samples, you are flying completely blind.
So what does this all mean for how we feed the cells? Why can't we just use an equation? Like, if we know how fast the bacteria normally grow, why not just program a computer to pump in the exact amount of food they should need?
Engineers absolutely tried that. It's an approach called simple exponential feeding. You set up a pre-programmed profile based on a formula.
Sounds logical.
In theory, they assumed a constant yield coefficient, meaning how efficiently cells convert glucose into biomass, and targeted a specific growth rate. The formula basically tells a pump to increase exponentially to match the cells.
But let me guess, a swimming pool full of stressed-out microbes doesn't follow a math equation.
Not even close.
Huh.
It fails on almost every level. First, that yield coefficient isn't constant. As the cells get stressed by those gradients, they start accumulating acetate.
Which is a toxic byproduct.
Right. Every bit of carbon they turn into toxic acetate is carbon. They aren't turning into healthy cells. So your assumed yield is suddenly wrong.
And what about the growth rate? Does that stay constant?
No, it drifts significantly.
Yeah.
These are heavily engineered cells holding plasmids, which are little instruction manuals we insert to force them to produce a drug. Curing those is exacting, so they might just drop the plasmids to save energy. Or they run out of a trace nutrient. The actual growth rate falls way behind the mathematical target.
So this open-loop system, is it basically like running on a treadmill that's programmed to speed up every 2 minutes, whether you're out of breath or not?
That is a brilliant way to picture it. The pump is completely blind. It ignores what the organism is actually experiencing. And the fatal flaw is it ignores oxygen limits.
The cocktail straw issue.
Exactly. At high cell densities, their oxygen uptake rate exceeds the oxygen transfer rate of the tank. The system is forcing glucose in, but the cells don't have enough oxygen to process it, so they suffocate. And because they're suffocating, they resort to partial anaerobiosis. They ferment the glucose, which causes a massive spike in that toxic acetate. If we connect this to the bigger picture, a rigid, pre-programmed plan lacking real-time feedback is just inherently doomed at scale. The biology is too dynamic.
Here's where it gets really interesting. We don't just have to rely on theory. There's a classic real-world horror story of exactly what happens when this blind treadmill approach fails.
The Konstantinov study.
Yes, the 1990 Konstantinov case study. It's the perfect illustration. They were running a 1,500-liter fermenter, trying to produce phenylalanine, an amino acid, using a recombinant strain of E. coli. E. coli 802471 to be exact.
And they started with standard procedure for the time. An uncontrolled baseline fed batch process with an exponential feed profile targeting a specific growth rate.
They set the math, turned on the pumps, and just let it run.
Yeah.
And by hour 18, it was a total disaster. The toxic acetate had skyrocketed to 30 to 35 grams per liter.
Which is an incredibly lethal environment.
Lethal. Cell growth stalled out completely at just 16 grams per liter of biomass. And the phenylalanine titer, a measly 8 grams per liter.
The batch was ruined. When Konstantinov's team did the postmortem, they found exactly what we just discussed. The tank ran out of lung capacity. The dissolved oxygen plummeted to a suffocating 5 to 10%.
And this was even with them pushing the equipment to the absolute limit, right?
Yes. Maximum agitation, 100% air saturation. The physical equipment simply could not transfer oxygen fast enough.
But the blind treadmill kept going.
Kept going. The glucose feed didn't respond to the oxygen demand. There was zero feedback mechanism to detect the overflow metabolism.
But they engineered a brilliant rescue. And this brings us to the hero of our deep dive, the balanced diostat.
A total paradigm shift.
They realized you cannot force a rigid plan onto a living organism in a chaotic environment.
Right. The balanced diostat is a model-independent, two-loop cascade control architecture. Model-independent is the crucial phrase. You don't need a perfect mathematical equation.
You don't have to guess the yield.
Exactly. The core principle is beautifully simple. You dynamically adjust the glucose feed rate to maintain the dissolved oxygen at a safe set point, typically between 25 and 40%.
So using our analogy, instead of the blind treadmill, this is like wearing a heart rate monitor wired directly to the treadmill. If your heart rate spikes too high, the treadmill automatically slows down.
That is exactly how it functions. The system uses two continuous loops. The outer loop acts as the brain. It continuously measures the actual dissolved oxygen using a probe, comparing it to the safe set point. If oxygen drops too fast, it generates a signal.
And sends it to the inner loop.
Right, the inner loop acts as the muscle. It receives the command and physically turns down the glucose pump.
But wait, if it takes a minute or two for the giant tank to even mix the liquid, doesn't the system risk overcorrecting, like slamming on the brakes too hard?
Great point. That's exactly why the inner loop uses lag compensation and anti-windup protocols.
Anti-windup.
Yeah, it's like cruise control for the pump. Built-in delays so the pump doesn't panic and drastically overcorrect, which would just cause the system to yo-yo back and forth.
Makes perfect sense.
Yeah.
And this is running alongside a few other automated safety nets.
Yes, parallel supporting loops. First, an agitation controller maintaining maximum sustainable mixing. Second, oxygen enrichment, which automatically injects pure oxygen into the sparge gas if regular air isn't enough.
Okay.
And third, an independent pH control loop. Vital. It adds base chemicals to keep the pH strictly neutral between 6.8 and 7.2.
Why is the pH so critical there?
Because if a tiny bit of acetate does form during a stress period, a neutral pH prevents it from turning into undissociated acetate, which is its most lethal cell-penetrating form.
Got it. Well, the results of switching to this Diostat system in the Konstantinov study are staggering. They ran the exact same 1,500-liter tank, same E. coli strain, but turned on the Diostat.
Outer loop controlling DO at a 30% set point.
Yep. Automatically choking back glucose if DO fell below 25%. The turnaround was miraculous. Acetate wasn't just reduced, it was maintained at absolute zero for the entire 36-hour run.
Zero.
The final biomass actually jumped to 36 grams per liter, a 2.25 times improvement, and the product hit 24 grams per liter.
A 3 times improvement over the open loop disaster. And completely reproducible across 6 consecutive batches. Though, to be fair, that level of reproducibility requires incredibly precise tuning. You can't just set the oxygen target at one number and walk away.
Oh, here we go.
No, seriously, the industrial guidelines dictate a very specific phased set point strategy. During batch growth, when you want rapid multiplication, you set the DO target at a comfortable 40 to 50%.
Okay.
Then during fed batch pre-induction, you lower it to 30 to 40%. This balances avoiding oxygen limitation while maximizing the feed rate.
And then comes induction when you tell the cells to start making the drug.
Right. Exhausting for them. Oxygen demand skyrockets. So post-induction, you raise the safety margin back up to 35 to 50%.
Wait, I have to ask about this detail in the data.
Yeah.
In the very late phases, when the tank is incredibly dense, like over 60 grams per liter, the oxygen target actually goes up to 40 to 60%.
Yeah.
Shouldn't it be lower since oxygen is so scarce?
It seems counterintuitive, but you push the safety margin higher because the physical limits of the massive tank, the OTR constraints, are completely dominating. If you don't force a high set point, that dense population will instantly strip the remaining oxygen.
Interesting. So how does the computer handle the physical delay of the tank when calculating these adjustments?
That comes down to the PID tuning. Proportional, integral, derivative.
The code.
Yes. Industrial recommendations are specific. Proportional gain is kept conservative, maybe 0.5 to 2.0. But the integral time is key, usually set between 5 and 15 minutes.
Because of the mixing lag.
Exactly. You have to literally tell the code to wait 5 to 15 minutes before aggressively overcorrecting. Otherwise, the computer reacts to ghost data from unmixed liquid. They also use feed limits of 0.1 to 1.5 times nominal and hard safety interlocks. If DO plummets below 10% for more than 2 minutes, a massive alarm triggers.
I just love how animated you get about the exact reaction times of this system. It's like you're talking about tuning a high-performance race car.
Well, it really is. Because when you tune it right, the ultimate advantage is that the Diostat is completely self-regulating. Axon and his team proved this. They took this Diostat architecture and ran it on 3 entirely different strains of recombinant E. coli.
Completely different genetics.
K12UL635, BL21DE3, K12UL634, vastly different metabolisms. But because the system just listens to oxygen demand, it delivered perfect control performance across all 3 without needing to retune anything.
That is an incredible engineering triumph. All right, let's take a step back and synthesize the journey we've been on today for you, our listener. We started by looking at the illusion of the lab scale.
A dangerous illusion.
Very. We saw how perfect lab conditions mask the harsh physical realities of a massive factory tank. The leap fundamentally changes the physics. Mixing takes minutes, oxygen struggles to dissolve, massive chemical gradients form.
Yep.
We saw how forcing a rigid, pre-programmed plan onto that chaotic environment inevitably leads to disaster, forcing cells to run on a blind treadmill until they suffocate in toxic acetate. But then the balanced Diostat.
The paradigm shift.
Exactly. Instead of forcing a plan, it actively listens and responds to the real-time biological needs of the organisms. By constantly measuring oxygen and throttling the glucose feed, engineers keep microbes in the sweet spot of productivity without pushing them over the metabolic cliff.
This raises an important question, though. We've spent decades engineering incredibly sophisticated control systems to force the industrial environment to accommodate the fragile limitations of these cells. Smarter feedback loops, better impellers.
Which brings us to a final provocative thought to leave you with today. Throughout this deep dive, we've explored how we manipulate the factory to serve the organism. But what if the future of scaling up biotech isn't just about building smarter sensors? What if, instead of fighting the physics of a 20,000-liter tank, we genetically engineer the microbes themselves to actively thrive in that chaos? Imagine an organism intentionally designed to love massive glucose gradients. A microbe that doesn't produce toxic acetate when oxygen drops, but just temporarily pauses its metabolism until it circulates back to an oxygen-rich zone.
Designing the organism for the factory.
Exactly. What if the next great leap isn't designing the factory for the organism, but designing the organism for the factory? It's something to chew on as you go about your day. Thank you so much for joining us on this deep dive. Keep asking questions, keep digging deeper, and we will catch you on the next one.
You've been listening to Biomanufacturing and Fermentation Technology with Prasad Ernala. Thank you for joining me. Until next time, keep engineering biology into reliable manufacturing.